Hi, my name is Sosica.
And my name is Anna.
We are postdoctoral researchers working in Queen Mary University of London in the Department of Material Science and Engineering.
We are analytical chemists and we are involved in elite sport research. So ESPRIT specializes in elite sport performance research. It is a big scientific project which consists of a multidisciplinary cohort with complementary skills.
Our lab is involved with a lot of biochemistry. Biochemistry is a study of the chemical processes in biological systems. We are part of ESPRIT, so-called O-team, which means Optimized Sensor Design and Embodiment, and we develop biosensors for continuous monitoring of physiological changes in the body of an athlete while they are training.
Keep going, you're doing really well. Come on, keep pushing, keep pushing.
A biosensor is an analytical device which converts a biological response into an electrical signal. Some of the sensor's operation is based on the action of the enzyme. Enzymes are naturally occurring, highly specific catalysts that speed up the reaction. A range of bodily fluids are monitored in the lab, including blood, tissue, and sweat.
A full map of physiological changes in an athlete can be produced. The information obtained by these sensors is used to aid coaches to improve the training program for the athletes and follow the progress. Information on lactate and oxygenation levels help to optimize the training regimes of athletes. For example, training at lactic threshold improves endurance.
During intensive physiological exercise such as running or cycling, oxygen consumption in muscles is enhanced.
Oxygen consumption increases as long as cardiorespiratory system is capable of delivering oxygen to muscles.
Doing really well.
VO2 max is the highest rate at which oxygen can be taken up and utilized by the body during severe exercise.
One, and stop it there. Feet on the side. That's it.
VO2 max value depends on many things such as gender and age, but most importantly it depends on the fitness level. During anaerobic, that is oxygen-poor training, lactate production is faster than lactate removal, which results in a lactate buildup in the body. Normal lactate levels in blood and tissue are very low. However, after intensive training and lactate threshold is reached, the concentration of lactate can rise rapidly. Lactate threshold is one of the most important parameters in training as it indicates the fitness level and it is the best available predictor of athletes' performance. Lactate used to be considered a harmful metabolite that deteriorates athletes' performance. However, today it's known that it's not the case. Lactate is also a primary energy source for the central nervous system. The lactate sensors we developed are amperometric sensors. We developed them on two different platforms. So this is one of the platforms. It is needle-based. There are two electrodes here integrated. So counter and reference electrode here are just one electrode. This is a stainless steel tubing, and inside there is a platinum wire which serves as a working electrode. On top of it there is a lactate oxidase enzyme which makes it sensitive and specific particularly to lactate. Advantage is that this can be implanted in tissue and it can be used for continuous monitoring of lactate for athlete running for longer time. We develop lactate sensors also on different type of a platform. On this kind of platform, which is a commercial platform with 3 integrated electrodes. Also here on the middle part in the working electrode we put lactate oxidase enzyme and some membranes which make it sensitive just in the range we are interested in.
The sensors need to have high selectivity towards the analyte of interest because other interferons present in serum and plasma get oxidized at the same time as the hydrogen peroxide oxidation reaction. This is achieved using suitable membranes. We are developing biochemical sensors for continuous monitoring of analytes like oxygen, glucose and lactate in tissue. We prefer tissue than blood because it's safer and also any adverse effects that can happen will be localized in tissue. Also, lactate and oxygen are related between each other, so simultaneous measurements of both of them will give a better picture for physiological status of the athlete.
Measuring athletes' performance from chemistry point of view is very challenging because bodily fluids are very complex, and also working outside the lab provides an environment which is complex and full of unexpected events. But this is also what is very inspirational, because we all the time have to keep learning new things, and this is what makes it also life of researcher very exciting.